So this is in continuous with my previous presentation or previous video that I have posted. You can see there that this slide shows you a YouTube link. This YouTube video has been uploaded in your Google Classroom also. So kindly go through the same. Okay, now talking about the modes of sequencing. The PacBio sequencing modes can be divided into two types, that is short insert and long insert. The short insert sequencing is called as circular consensus sequencing, whereas the continuous long read sequencing is for the long inserts. What does that mean? So this means that if the template size is comparatively small, when we say that small means that it is smaller than the longer inserts, or the small does not mean as small as for the Sanger sequencing or as small as for the Illumina or the 454 or iron tolerant sequencing. The small here ranges from 100 base pair till 300 kbs, 3000 bases. So that is the length of the short reach by definition of the PacBio. This kind of sequencing is not possible in the earlier platforms. Okay, so what is the advantage of doing the circular consensus sequencing? The advantage of circular consensus sequencing is that we know that our sequencing platform or PacBio platform has an error rate. In order to minimize that error rate, what it does is it sequences the same molecule again and again in a circular fashion. Why is it possible to sequence in a circular fashion is because you can see the two templates or bell-shaped adapters are attached to the two ends of the DNA. When the two strands of the DNA get separated from each other, then it makes a circle. And the polymerase can go round and round this circle n number of times to sequence the same molecule again and again. If we sequence the same molecule again and again, it is going to give us, and the sequencing, if we align them or if we put them one over the above, then we would get the consensus sequence, which would tell what would be the exact sequence of that particular insert. So circular sequencing is called as consensus sequencing because after sequencing n number of time, the consensus sequence is taken as the final sequence for a particular insert. However, if our aim is to sequence longer reads, then this circular consensus sequencing is not possible. Okay, so you can see here, this is a double stranded insert, and when it goes for CCS sequencing, you can see that the DNA overlaps and the complementary strand is, as the polymerase moves down, this is polymerase which is moving down and the DNA is being synthesized, and this process is repeated again and again, and whenever it is repeated again and again, the sequence is recorded in case of circular consensus sequencing. So this slide had this video, but I am not putting this video over here. Again, this video you can see in your respective Google Classroom slides. Okay, now I would want you to see this video for circular consensus sequencing.
Circular consensus sequencing, or CCS, is a highly customizable PacBio sequencing application that generates consensus sequences from multiple passes around a single circularized DNA molecule. The polymerase used for sequencing passes multiple times around the circularized fragmented DNA. When there are multiple polymers, strands will be inserted upright into it. And these multiple passes will separate
So what I want to tell you is that in the circular consensus sequencing, you can see that as the polymerase is polymerizing or adding the nucleotides, the double-stranded DNA becomes circular. And in the circular fashion, we can set up the sequencing machine in such a manner that it can go around the same template again and again n number of times. The number of cycles that we want the polymerase to go around, it depends upon the what is the length of the uh, insert. If the insert is very long, the number of rounds or circles will be more. If it is shorter, we can keep it to minimum. The advantage is that with this help, the circular consistent sequencing can be used for metagenomic population. It can be used for minor variant detection, and in case if the samples are on a large scale and the sequencing them on a Sanger platform is very expensive, then alternatively PacBio sequencing can be used to carry out high throughput sequencing of those products which are whose sequencing is on a costlier side or expensive side on a Sanger sequencing platform. So that is the advantage of it. Okay, now moving to the next slide. So coming to the second type of sequencing mode, it is the continuous long read sequencing. Continuous long read sequencing does not allow the DNA molecule to go in circles. Okay, why it does not allow is because the length of the insert is very long, and as a result, it is not possible to complete the circle so easily. And with as the DNA molecule, it goes, as the length of the insert increases, the sequencing error also increases. So if we want to sequence the genome of an individual or a eukaryote, or in that case, so continuous long read sequencing is preferred. Or if your insert size or your sequence in question is requires the need of very long sequencing, then the continuous long read sequencing is done. So coming to the platforms of PacBio sequencing, the PacBio sequencing platforms can be divided into different categories. In 2010, the beta version, and in 2011, the commercial version of PacBio sequencing platform was RS system, which was later on evolved into an RS2 system. You can see over here that from 2011 onwards, there have been so many variation till 2014, and you can see that in these different types of variations, you can see that to start with they had C1, then they had C2, P4XL, P5C3, P6C4, and all of these, they differ from each other in the average read length and the amount of data they were delivering. Okay. However, with the advancement after 2014, in 2015, the company again came up with another system which is called as the SQL system. The SQL system, the advantage was that in this case, the SMRT Cell had 1 million ZMV holes. That means the capacity was tremendously increased and the average lead length came from 10 KB till 100 KB, and the data which was generated was also humongous. Okay. So what was the advantage of the SQL system over the RS systems? It was that in comparison to the RS system, due to the increase in the ZMVs on the surface of the SMRT Cell, the throughput was increased almost 7 times. The accuracy also increased to 99.999%, and the length of the average reads also increased till 10 KB. for continuous read, not for circular consensus read. For continuous long continuous reads, the lead length went up to 80 kb. Okay, with 99.999% accuracy, and the average read length went up to 20 kb. Since it did not involve PCR amplification bias, therefore it's evolved as a very good technique to study the DNA modifications and the DNA variations. In 2019, specifically April 2019, PacBio has come up with another technology called as the 8M chip. 8M means million chips. So as the name suggests, here the smart cell, instead of having 1 million ZMVs, now it is having 8 million. So therefore the throughputness has increased by the factor of 8. Okay, and the amount of data that people are getting, and also the read length and its accuracy has also increased. The sequencing system always keep on evolving with the as the time passes by, and the error rate also keeps on decreasing. This slide gives you an overview of the difference between the PacBio smart sequencing and Oxford Nanopore smart sequencing. The two differ in this, that in case of PacBio sequencing, you know that both the cases there are holes, but in this case the DNA does not pass through the hole. In this case the DNA pass through the hole called as the nanopore. Here the DNA is restricted inside the ZMV in an illuminated volume, which is called as the illuminated dissection volume. So that is the difference. Here the nucleotides are fluorophore labeled, therefore fluorescent signal is produced. The nucleotides here are not fluorescent labeled. However, the electrical signal is produced when the nucleotide they bring about the change in current inside the nanopore. Here the fluorophore signals are recorded, whereas here the electrical signatures are recorded for the nucleotides. So this slide gives you some additional YouTube video links which are there out there on the YouTube channel. Kindly go through it, and kindly I have also attached a PDF file showing the history of the PacBio sequencing. Kindly go through it. And I hope all of you are safe, you are healthy, enjoy. God bless you all and take care of yourself. Thank you.